Great, thank you so much to MDC for inviting me to be here and to speak today. I'm going to try and tackle a bit of a clinical trial update for Duchenne Muscular Dystrophy. And first here, I do have a relationship with many pharmaceutical companies, primarily as a site investigator, but I have chaired PTC Advisory Board for Canada, and that's particularly relevant to some of the data that I'm going to show here. And I do have some investigator-initiated grants from several companies as well. I serve as a Data Safety Monitoring Board member for Catabasis and Solid. That's money that I do take personally. The rest I donate to charity. So we're going to try and fit in 30 minutes what is a very rich and exciting area of novel therapeutic development. And so I'm going to try and take mostly examples from trials that I've been involved with or that have been particularly relevant for Canada or particularly controversial or important for us to know about. And we'll move through them. I've divided them into more genetic-based therapies, muscle modifiers, and secondary injury is how I sort of think about them. And so if I've missed something, of course, happy to try and field questions about it or talk about it later. But I'd like to start just by showing again the dystrophin protein associated complex here that, I don't know that, oh yeah, that I think we're all familiar with in this room. So dystrophin sits as a large protein here that binds the intracellular cytoskeleton to, whoops, my apologies. That binds the to the muscle cell membrane. Oh my goodness. Okay, no pointer. I'm gonna. And that links to the extracellular matrix. And even though we think of dystrophin as a structural protein and maintaining a scaffolding or structural integrity to the muscle cell itself, dystrophin has many other roles, and I won't get into them here, but I think the downstream effects of dystrophin loss allows us for many avenues for therapeutic intervention, and obviously we'll talk about some of those today. One that I'd like to shed a little light on just before we dive into the specific therapies is its impact on satellite cells. And this is, I think, exciting work that's had a bit more of a light shone on it recently from Dr. Rudnicki. And really what he's shown in a 2016 paper is that the polarity of muscle, maybe I'll try this one more time just to show you. So in the muscle milieu, we have a set of satellites, sort of stem cells, that are involved in regenerating the myoblast and eventually the myofiber population. And those stem cells have to divide. And you can see that process here. And it's really important that they have the proper polarity to divide. And once they divide into a muscle progenitor cell and then a stem cell that's going to stay there as for future propagation. But in the case of dystrophin, actually plays an important role in that cell polarity. And so often when this process doesn't go well, the cell ends up going back into a senescent state. And contrary to, oh, thanks so much, perfect. And contrary to probably one group of people, the hypothesis about dystrophinopathies where the stem cell population gets depleted, in fact, we're recognizing that that's not in fact true. In fact, there's a higher level of senescent stem cells that sit in the muscle milieu, but that appear that they can't be activated to regenerate this this muscle progenitor pool. And that's very interesting, because I think this sheds a bit of a new light on how we think about these muscle progenitor cells and our novel therapeutics. How are they going to impact? So, for example, will utrophin upregulation actually replace dystrophin in this kind of scenario and help us recover these muscle progenitor cells? Or AAV9 viral-based vectors, they don't actually infect muscle stem cells very well. So things like this, I think we need to think about as we move forward. And I've put a couple of references here. One is to this very interesting paper by Vieira around the discovery of this JAGED1 gene that when upregulated in the muscular dystrophy model in golden retrievers, actually improves this dynamic, improves transition from senescent satellite cells to muscle progenitor cells, and largely rescues the phenotype in that animal model. And so these are important things that I think we'll hear more about as we think about therapeutics for DMD. So I'm going to go into therapy update, but before we do that around novel therapeutics, we obviously have to remember and recognize the importance of good clinical care in the health and life of patients who are journeying with Duchenne muscular dystrophy. And I want to point out that the new care guidelines are out in the Lancet Neurology. They're a three-part diagram, and I'm not going to go into the changes right now from the last document, but these are important reads for anyone in this field, please. The one thing that is lost in the new care guidelines, which I'm disappointed to see go, was this diagram, which I think really represents, how we should care for people with DMD and their families, with them at the very center, and care coordination, and then all the other, sort of more medical modalities that we think about. And I always love this diagram and try and keep it as guidance. And so let's not lose track of that with all the tables in the new care guidelines. So let's go on to the genetic-based therapies. And as I said, I'm giving this overview slide, and then I'm going to go into one particular story around Adlorin. But of course, exon skipping, very exciting. We do have a drug approved in the US, eteplirsen, and we are treating some kids in Canada still with drisapersen who are involved in that BioMarin study, and of course about to launch exon skipping trial with Sarepta for exon 45 and 53 in several sites in Canada. What's interesting to see is the next generation of exon skippers are coming along, and Sarepta and Wave both have a next-gen exon skipping for exon 51, and we will definitely be doing this. It's a phase 1 single-dose trial, and I believe we're looking at options for this in Canada as well. And then to skip down, of course, gene replacement itself is fascinating, has certainly had some hurdles already with studies nationwide and some of the unexpected immune reactions there. But now, Pfizer's starting a phase 1 trial, and Solid Biosciences has dosed one patient, as many of you will know who are following this story. And although it can't be discussed more publicly, I do sit on the Data Safety Monitoring Board, and happy to say that the patient is is now doing well. The study's on clinical hold as we work through a new safety plan with FDA. So I'm going to talk a little bit about Adaluren. I think everyone's aware of this medication. Adaluren has been in clinical trials since 2008 in Canada and around the world. And it's a nonsense read-through drug. So for those 10 to 12% of patients who have a premature stop codon in the dystrophin gene, this molecule binds to the ribosomal machinery and allows you to read through this while still respecting the normal stop codon at the end of the gene. And I'm not going to go into these in great detail, but I do want to show you, I'm going to show you the meta-analysis data. So I want to show just for people who don't know, there's been 2 well-designed large clinical trials done, a phase 2B study, which the design is here. This was the one in 2008. Unfortunately, very close to reaching statistical significance, an important clinical difference here in the results. This is the 6-minute walk test across 48 weeks. And then this was followed up by a second clinical trial called ACT-DMD that was enriched to try and make sure that we had a more responsive and less variable patient population. But probably more important in this study as well was some pre-specified analysis as our understanding of the 6-minute walk test became better. And so the focus was on this baseline 6-minute walk test of 300 to 400 as maybe the group that we could see the best response on this test. And so I want to make that clear, not necessarily the best response to the drug, but the best response on this test over a 48-week time period. And you can see here again, unfortunately, the overall study didn't reach statistical significance, still showed a benefit for Adaluren. But in this pre-specified group between 300 and 400, you can see a dramatic difference between Adaluren and placebo in terms of benefit for 6-minute walk distance. So it seemed like the thinking around this subgroup was relevant. And so this is, looking at the meta-analysis has been particularly important for me, and I've been pushing, PTC for a while that that for the clinician this is really relevant. And these tables come from a paper we've just submitted, but the data has been presented at several meetings, so this is in the public realm. I just put the tables in because I like this formatting better of how we did it in the paper. But what I want to show here is that, for clinicians, when we take the evidence and combine it together in meta-analysis, I don't think there'll be much argument that meta-analysis is really the highest level of evidence we can get. What I show here is the results from the intention to treat patients in the phase 2B, the ACT-DMD trial, and then the meta-analysis. And I've done this with the 3 different subpopulations. I'll be honest, I'm most interested in this because this takes all comers from both trials, puts them together, overcomes the difficulties of doing rare disease trials where you have small numbers of patients, a lot of variability, you don't understand your outcome measures as well yet, and you can see when you take all comers that Adaluren does have a benefit of just under 20 meters difference, and the differences obviously become more dramatic here. And just to outline the consistency of the results as well, so even though the focus was on the 6-minute walk test as the primary outcome. Of course, these show the timed functional tests, so 10-meter walk run here, the 4-stair climb and descent, and the trends are the results are all here very clear as well as we combine those together. So definitely that, for me as a clinician, thinking about evidence in the GRADE framework, this is compelling stuff. The other thing that we've been looking at recently, and this will be submitted shortly, is a comparison between the open label extension data from PTC 019 to the Synergy control group. And so I put two graphs here, one on ambulation and one on forced vital capacity modeling. And what you can see here is, again, compared to, sorry, these are very small legends, but here's Adlorin treated group in red and the control group from Synergy. And you can see a difference in ambulation. And at the 50th percentile here, there's about a 1.2 year difference between these two groups. And if you model FVC decline as well, what you can see here is about a 4-year difference between the Adlorin treated group that's down here. So a bit confusing because they're in green, but the green here is Synergy. This is the Adlorin group versus Synergy data. And so it appears to have some emerging evidence around prolonged ambulation and pulmonary function that one can't, appreciate necessarily in a shorter 48-week trial. So let's talk a little bit about the muscle modifiers. This is a very crowded space, lots of things going on. I'm going to focus a little bit on myostatin inhibition because there's lots and lots of people in this space, and it's a very exciting area. But we're doing some work with Jacques Tremblay, who's here, around myoblast transplantation. Utrophin upregulation, I think, is really something everyone's excited about. I hope we'll hear shortly from Summit about their phase 2 study. Those the 1-year follow-up from their, I think, 40 or 50 patients in the phase 2 is due sort of now. So we should see that. There's a number of antifibrotics that are in play here, particularly givinostat. And again, a lot of these things, I think, converge on similar pathways, and I won't go into it, but it's very interesting to look at the antifibrosis, the effect on satellite stem cells of all these products. So that's important. And I've been very interested in this contractile optimization. We're doing a study in SMA right now with this cytokinetics drug, but I still think it would be very interesting to look at in muscle disease. So I think myostatin inhibition is familiar to all of you, so I won't go into great detail, but myostatin, which I've shown here, is a peptide that circulates and binds to the activin 2B receptor and signals a downstream cascade that basically suppresses muscle development in a way. And if you're born without myostatin, either in an animal or human model, you have large muscle hypertrophy, relatively strong. And the good thing is that myostatin doesn't have a lot of impact on other tissue like heart or smooth muscle. And so you're relatively healthy despite this muscle increase. There's not a lot of off-target negative effects of this. And so of course this has become an important target for people. People will remember that we started this work, did the first trial in Canada here with a myostatin inhibitor and DMD with ACE31. Although we had very good results, and you see over here the treated groups in red and green had maintained 6-minute walk test versus placebo, and we showed improvements in lean body mass and bone density and fat mass. We ran into some difficulties with telangiectasias and nose bleeding, and eventually this product was terminated, but I think it was important first work, and a number of my Canadian colleagues we've published this a couple of years ago. So this has resulted in a number of follow-on products, particularly from Acceleron, which have developed a local injectable myostatin inhibitor, and we're currently trialing that in FSHD, in biceps and TA, but I really think it would be very interesting to look at this in DMD as a local based therapy. I'm excited about that. This shows some data from the healthy volunteers that were in the phase 1 study, but actually data from this FSHD study was just presented at AAN last week, and does show improved muscle volume and fat content. And of course, there's 2 trials that have been active in Canada with myostatin inhibition, one with Pfizer, which uses an antibody directed against myostatin, and then a study by BMS, now acquired by Roche, that uses a slightly different biologic, a nectin-based chemistry to bind myostatin. And so both of these are ongoing in Canadian centers, and if you're interested, please let me know. So just looking at secondary injuries here, a number of things in this category. We've looked at tadalafil for DMD. Unfortunately, that was a negative trial. Looking at vascular reactivity, there's calcium modulation, trying to reduce calcium influx into the muscle cell, and this is in this remepramide was in a phase 1 study, and then of course Santhera has a study pathway with looking at ademodone as an antioxidant. I'm going to focus just very quickly on the anti-inflammatories, because of course, you know, this is, so if you can't see this, corticosteroid management. You know, this idea of reducing inflammation has been at the center of DMD care, and in fact is probably still the only therapeutic that has really moved the yardsticks in terms of important outcomes for DMD. And yet a lot of questions remain about steroid treatment, and so, you know, what's the proper mechanism, the best regimen, you know, what are all the side effects? We're starting to see some data, more data come out, and I'm going to show you a study we're doing right now. But of course, Birch Griggs published a recent study in Neurology that was done 20 years ago, never published, that showed a comparison between different regimens of steroids, so 2 doses of deflazacort, prednisone, and placebo, and showed a good improvement in the steroid-treated groups and not so much in placebo over a 12-week period. And then over a full year, you can see the 2 doses of deflazacort versus prednisone here, and again, you know, significant improvements over that time period. So this was published not long ago, and I think is helpful to inform us as we're doing the 4DMD trial, which is an international randomized controlled trial comparing 3 different regimens of steroids, which I think will give us lots and lots of answers about what the best regimen is, when to start. We've got a broad range of kids in that study, and really a robust list of outcome measures. And so enrollment's complete for that, and that's a 3 to 5 year follow-up plan for that. But even though we're trying to understand the impact of steroids still, and many of the big questions about it, there's been a widening of thought around trying to decrease the inflammation in DMD, and yet spare kids from some of the steroid side effects. And so a number of groups and companies are now focused on inhibiting this NF-kappa B pathway that appears to be particularly relevant in DMD. The NF-kappa B pathway is activated early in the disease, and we have animal models that show that NF-kappa B creates a phenotype that could be consistent with some of the features of DMD, and again, you know, this cascade of inflammatory processes really have probably widespread effects. 1 minute, yeah, okay. So, but I got 2 slides. But what I don't want people to forget is just, you know, making sure we know what we're trading off potentially for this inflammatory stuff. And I think we need to be thinking carefully about whether there's a biomechanical advantage to being short. There's some data around that, but we haven't fleshed that out very fully. I've shown some data here with kids who are of normal height versus low or very low stature on ambulation. This is Canadian data that we've presented, and there's about a 1-year difference in ambulation if you're short or very short compared to if you're normal stature. And of course, if this is true and robust, we're actually doing this internationally now through the registries, then this is important data for us to understand. I was going to talk a little bit about catabasis, but that's one of the companies doing this. So I'm going to stop here and just recognize that we've got lots and lots of pathways to impact on this dystrophin deficiency that we deal with in Duchenne and Becker's. I think ultimately one would love to see a pathway forward that exploits multiple, points in this pathway in combination, and of course I didn't even get into, but it's a massive topic in and of itself, is the challenges in clinical trials and regulatory pathways for new therapies in DMD that we all have to be in the ring together. So I thank you again and appreciate that. I mean, aside from going back through the various mechanisms, I mean, some might have impact, like just by their biologic plausibility, but in terms of, sort of actual outcome measures from clinical trial, to be honest, we know very little. I mean, in fact, as you'll know, we even have, it's very little good evidence about even cardioprotective, like let alone the novel therapeutics, even the cardioprotective drugs. But I mean, to my knowledge, cardiac outcomes have not, I mean, please, anyone in the audience who can, I mean, there's really not cardiac outcomes that have been part of primary or secondary outcome measures. And part of it's too, well, I mean, there's several reasons. I mean, one, as you can probably see from what I've shown, most of the trials for novel therapeutics have been done in young children. That we're still struggling. I mean, I didn't even, about how to address and design clinical trials that we can look at effect size for, for multiple outcomes, upper extremity, lower extremity. And so a lot of the trials have been on young kids. So of course, we don't really see clinically relevant cardiomyopathy in young kids anyway. So that's part of the limitations in and of itself. Yeah, I genuinely believe this. And in fact, I think local therapies should stay in the game. I mean, not that I want to sound unhopeful, but we've yet to really hit a home run here, folks. And so to me, it makes a massive difference for a boy to be able to use their wrist extensors or grip their video game. That's actually life changing for kids. And so the critique against this, I think is, I don't think takes into account really possibilities for quality of life and individual goal setting for patients, which is why I'm so interested in cell-based therapies as well for local, for really trying to overcome local limitations. But I'm open to contrary thoughts. Thanks.